Hello. This is one heck of a chapter. Tons and tons of stuff. It'll be much easier for you guys doing this with an open book test than regular classes that have to memorize a lot of the stuff. But hopefully, you will still learn a lot of information. Now, neuroscience is my jam. This is what I do. And again, this is how I met Mark Breedlove long ago. And as always, he has an opening vignette, which he will refer to during the chapter. Um, you don't have to know this, but Deidre, who laughed when they stimulated her brain, she confabulated. She made up reasons for her laughter, even though the reason for her laughter was direct electrical stimulation of her brain. So this is the machine. This is our essence, uh, brain and what's connected. So there's chapter preview, and neurons are very important to us. Glial cells are very important to us. Glia. I don't think they get a lot of coverage in this chapter. They've been becoming more important in recent years. The next edition of this book, I'm sure, will reflect the importance of glia. And there's, your simple cell on the left, membrane, cell body, or soma, uh, nucleus, the purple thing. And there's the stereotypical neuron we use to teach everybody what a neuron looks like. There's the cell body, also referred to as the soma. The nucleus is in there. Dendrites. I remember being amused that when my daughter was in early grade school, that they would go out and look at tree dendrites and characterize trees. And once you see the connection between dendrites and tree branches, you can't ignore it. And there's a common axon, and then there's the ends of the axon, the dendrites, or we used to call them telodendrites, but now we don't tend to do that. And those little swellings at the end are what we'll see, stereotypical representations of synapses, but synapses can be much more complicated than that. Oh, there's glia. There they are. Hello, little guys. The name glue suggests what we thought they were for, but oh my, they are more involved in neural functioning than we thought. We have upwards of 50 to 100 billion neurons. Each neuron, on average, has thousands of synapses. There may be 10 times as many glial cells as neurons. It's complicated in there. It's been estimated that there are more potential neural states, not neurons, neural states in a human brain than there are molecules in the known universe, not the solar system, the universe. It's complicated. Synaptic cleft, there are cleft chins, there's a little crease there. Synaptic clefts are little gaps, and they're important for synapses. Vesicles are like little water balloons. They often contain neurotransmitters. Neurotransmitter receptors are very, very, very important. You really have to get your heads around how chemical neurotransmission exists. Now, he makes a point here that information passes in only one direction in a synapse. Actually, there are often bidirectional communications where the cell that's receiving information also sends information back to the original cell. Schizophrenia will come up in this chapter and quite a bit in the next chapter as well. There's the number of synapses, and each synapse can have a number of electrical states. So it's not just 10 to the 15 synapses, it's 10 to the 15 synapses times number of states each of those synapses can have. It's beyond comprehension, which means neuroscientists will have jobs for a long time. Neurotransmitters transmit information across synapses. Amen and amen. And there are some nice figures. And you can see how that mustard colored cell is sort of draping synapses all over the neuron it's trying to talk to. And in real estate, they often say location, location, location. Where the home is important to the home value. And where synapses are important. And so here we have synapses on far away or distal dendrites and proximal or close dendrites, and also right on the soma. And some synapses occur right down here where the axon starts. And they form a pseudo-democracy, sort of like our democracy. If you can afford lobbyists to go to Washington, D.C. and speak directly to your representatives, you have more power. And synapses down here by where the axon starts are more powerful. There's a little picture of the synaptic vesicles. I usually talk about these as being like water balloons, as I think I've already mentioned. If you throw a water balloon at a screen door, the water balloon, the membrane stays inside the house, but the contents of the water balloons spill out into the synaptic cleft. And there are little blue guys on the receiving cell. That's the neurotransmitter comes from the presynaptic membrane. It goes across the synaptic cleft and it hits the receptors on the postsynaptic membrane, and then it has to go away. And sometimes it goes away by going back into the original cell. Sometimes it just floats away. Sometimes there are enzymes in there that destroy it pretty quickly. And here comes the small array of neurotransmitters. Acetylcholine was discovered first. It was part of research that led to someone getting the Nobel Prize. And there's an important truth here. Acetylcholine on the heart slows the heart. But I'm talking to you using acetylcholine. You flex your muscles using acetylcholine. So the same neurotransmitter can have very different effects on different receptor neurons. Glutamate will come up a lot in the course, and it's used everywhere. And it's almost always, as it says, excitatory. Acetylcholine is more complicated. And there's a table. There's acetylcholine. There's norepinephrine, which tends to speed up heart rate, balances off acetylcholine on the heart. Glutamate, we've mentioned. GABA, gamma amino butyric acid, very important inhibitory neurotransmitter, often interacting with glutamate. Serotonin, important for mood and thought to be involved in depression and pain, but it's complicated. Dopamine, often said to be involved in reward. I like to say it's involved in vigilance, to tell you something's important. And my research life was spent with the endorphins. which can reduce the perception of pain. And I am a big fan. There's reuptake, which I alluded to on the last page. Often, but not always, neurotransmitters are taken back up into the axon terminal that released them. And if you block reuptake, of course, the neurotransmitter stays in the synaptic cleft longer and can affect the postsynaptic membrane more. The still popular antidepressant drug that we use is a selective reuptake inhibitor for serotonin. The trade name is Prozac. And it's not as magical as I wish it were, but it is popular. So with Prozac, you're not directly stimulating a postsynaptic receptor, but you're allowing your body's own voice to be more impactful. And there's motor neurons, which I'm using to talk to you, and there are 3 different types of neurons there. 2 of them look largely alike. You have the one we taught you with, and then you have a bipolar one, which has 2 poles coming off the soma, and they're common in the sensory systems. And interneurons, the most common neuron in the body. They're like virtually everywhere, and they look like the standard neuron that we teach you with. And notice that there's a little halo around where the axon comes off the soma. That's where the action potential starts. That's where information starts down the axon, which is why I mentioned lobbyists, because if you can have synapses near there, you can really affect whether anything goes down the axons. And reflexes, we have on the next page. This is a common one, the hot stove reflex. This is related to pain. This is something that I'm involved in. One thing I would complain slightly about here is you see this swelling here. Well, there should be a neuron there stuck off that axon that looks like this one. Although there's only one branch, there's only one axon coming off of it. The axon bifurcates, splits, and part of it goes out to the skin and part of it goes into the central nervous system. So it's not a fatal mistake. It's just, it bothers me. It should look like a little lollipop sticking off that axon. And then you have a synapse in the spinal cord talking to an interneuron. Now notice the interneuron is just a ball, but we know that they don't look like that. They look like the picture on the other page. And then the motor neuron also doesn't look like a ball, but they look like the neuron on the other page. And so you have in this reflex 2 synapses in the spinal cord, one between the sensory neuron and the interneuron, and then a second one between the interneuron and the motor neuron. There's also a third reflex that if it doesn't exist, you're in trouble. And in fact, if these synapses start being attacked and they die, so do you, sadly. The synapse between the motor neuron and the muscle. And by the way, acetylcholine is down there. Acetylcholine receptors are on the muscles. So that's a withdrawal reflex. Neural networks, we've got so many networks. It's so complex in there. And here comes the soul, which means we're coming up on Descartes. He was a mind-body dualist. And here we go. So there's artwork. I took a course at UCLA on the history of artwork in neuroscience. Descartes may or may not have drawn that, but he thought that there were little tubes that carried information up to your brain, and a lot of what we do and a lot of what other animals, all of what other animals do, is reflexive. But he thought the soul, mind-body dualism, the soul being the mind, interacted with the body at the pineal gland, and so he was able to retain the mysticism, believing in souls, but allowing them to interact with the body in the pineal gland. He thought that only humans had the pineal gland. If he was right, that would have been nice, but he's not. Other animals have pineal glands for sure. I have a little cyst on my pineal gland, which hopefully is still not growing. Body electric. Now we're going to do action potentials, which I usually don't spend much time on because most of what we do doesn't affect these. By the time you affect action potentials, you're in real trouble. But there's a multipolar neuron, and then you've got a voltmeter inside, and it's telling you that inside relative to outside is about 60 millivolts. Milli is a thousandth. That sounds like a small voltage, and in a sense it is. But in a cell that small, it's quite a big voltage. And there you have a pump that is pumping positive ions out in that picture. And I won't go into any more detail on that, other than to say that that pump requires a fair amount of energy. And doesn't work, we don't work. And the resting potential is what the neuron is at when it's not generating action potentials. And this book says minus 60. Sure, why not? It varies across cells. And there's a cell membrane, which we really haven't talked about, but it's got two layers on the outside, and on the inside you have fatty layers that like water, and in the middle you have areas that don't like water. And that channel is transmembrane. It's giant. It goes from outside the cell to inside the cell. It's made up of many proteins. It's got probably multiple receptors on it. And you could spend your life, and people do spend their lives studying that. Potassium is important. Sodium is important. They both have positive charges, which is confusing because you're going to think that there must be positive and negative charges moving across the cell membrane, but positive charges going in make the cell positive inside. Positive charges going out make the cell negative inside. So everything in the action potential is actually positive charges moving in or out. And there we go. I teach behavioral neuroscience. This textbook, that textbook is published by the same company, and this graphic is in there as well. Don't spend a lifetime looking at this. It's gobs of information. The concept of threshold is important. This is like lighting a fuse. It doesn't make any difference how hot the stimulus is. If you can get to the threshold, you get the action potential. And it goes positive and then it goes negative. Both of those are based on positive ions. Going positive means, because we measure inside relative to out, positive ions go in, and then when it goes negative, it's negative ions going out. And that hyperpolarization there is important, but we don't deal with this very much. Again, I often skip virtually all of this in my live course. And the action potential is like a burning fuse. So I like that analogy. It's not like a bullet being fired. If you've been taught that, it's a dangerous concept. There's everything I just mentioned. They go down the axon, almost always. And there's voltage gated sodium channels. Those are the ones that cause that spike, the upward phase. And there's sodium opening and going down the channel. This is a dead zone because the channels won't reopen for a period of time after the action potential comes through. So it keeps the action potential moving in only one direction. If you came down here and started the action potential, it would move in both directions. It moves in one direction normally because of that uh, what's called the absolute refractory period. Once you're in the absolute refractory period, it's absolutely impossible to generate another action potential until those channels reset, in a sense. And I taught out of this book before, and there's your toilet, and not a fan of this. I don't teach using it, but it is useful when it talks about refractory phases, and you have to refill it. I don't know. I'm not in love with it, but there it is. And there's axons and dendrites and refractory phases again. There's really two. The book only talks about the absolute. Myelin speeds up action potentials. And myelin is not part of the axon. It's placed on the axon. With multiple sclerosis, your immune system starts destroying myelin in your brain. I had a condition where my immune system destroyed myelin in my peripheral nervous system. That was quite an adventure. Apparently, I didn't die, but it was a good one. So there's myelin. It's like insulation on a wire, and the voltage-dependent channels are at the nodes of Ranvier. I forget whether this book gives you that title. Yeah, there it is, nodes of Ranvier. Notice Ranvier is capitalized, suggesting it's a human being, because it was. And there we go. And if you have MS or what I had, the not having myelin doesn't just slow things down. The action potential doesn't get through. Notice that there are no channels here. And if this myelin goes away and you have exposed wire with no channels, the action potential usually can't even get through there. Saltatory means leaping or jumping. It's a nice word. We had a faculty member who had my condition too as an adult. That almost killed him. I was not hospitalized. So there's the life story of a neurotransmitter. Action potential comes down, synaptic vesicles fuse with the presynaptic membrane. They don't leave, they just dump their contents. They hit receptors. In these cases, there are sodium ions, but there are other kinds of receptors that actually aren't channel receptors. And then there's reuptake and recycling. And here's an enzyme that can destroy the neurotransmitter in the synaptic cleft. That's what happens with acetylcholine. But many of the neurotransmitters that we're interested in psychology are recycled, like serotonin and Prozac. We don't recycle Prozac, but Prozac affects that recycling. So that's what happens at the end. And Nobel Prize for my friends. There's Lowy. He had a dream. Actually, he had it twice. The first time he forgot. And there's a frog heart in life-sustaining buffer, and the vagus nerve is wired up to it. And the vagus is becoming very big these days. You electrically stimulate that when it's connected to the heart. The heart slows down, as you can see in that figure. And sort of miraculously, some of the acetylcholine that's released from the vagus goes into the liquid. If you suck up the liquid and put it on another heart, it slows down too. Thank you for the Nobel Prize. So Lowy, acetylcholine is the first neurotransmitter that we were able to effectively study, in large part because it was available in the peripheral nervous system, like innervating the heart. And up until Lowy, there was a huge debate about whether synapses were electrical or chemical. And certainly the ones we're mostly interested in were and are chemical. And drugs and the concept of agonists, which activates receptors, this book says. I like to use the term agonist to refer to increasing the effectiveness of a neurotransmitter, which can be done in the way described here or otherwise. And antagonists, and he talks about binding to the receptor. I would say anything that disrupts the normal neurotransmitter is an antagonist. We're not arguing. He's just simplifying to make it so it doesn't hurt your head. So an agonist acts like the neurotransmitter at the receptor site. You don't need the neurotransmitter. An antagonist blocks the receptors for the neurotransmitter. And I've done a lot of research with naloxone, which is used nowadays to save lives after opiate overdoses. I can't tell you how much research I've done with naloxone. It's a receptor antagonist. It's a very safe drug, by the way. Heroin acts on receptors, and naloxone, which I'm not telling you to know it, you'll know it later, unless it's already in the book, I forgot. And then alcohol affects GABA, and for some reason my students in neuroscience are very happy to learn that. And when I ask them to talk about neurotransmitters, they almost always pick alcohol and GABA. GABA is very important neurotransmitter. Remember, we've already said it's inhibitory. Hormones, by the way, Mark Breedlove, your author, his specialty was and largely is hormones, and the pituitary is important here, which is not the pineal. Pineal is Descartes, and that's where the soul, if you're a dualist, controls things. The pituitary is often referred to as the master gland, and oh my, it does stuff, and releases many hormones. Just appreciate that. Lots of things. And again, the book is emphasizing receptors that are channels. Not all receptors are channels. And there's the concept of master gland. And we would be in trouble without the pituitary for sure. And there's a discussion. And I love anatomy, neuroanatomy. You have to look carefully. So blue is the central nervous system, which is the brain and spinal cord, which is not the skull and spinal column. Those are bones. Now the brain is easy to see. And the spinal cord is coming down here, and it's coming down, it's coming down, it's coming. Oh my goodness, it stopped. It's if you can touch the upper part of the curvature of your lower back, which I can't anymore. That's where your spinal cord ends. Nerves continue down through the spinal column here, but they are no longer central nervous system, they are peripheral. By the way, if you get a spinal tap, they often go down here because the giant needle they use then can't hit the central nervous system. And there's the nervous system: peripheral, somatic, autonomic, sensory motor, sympathetic, parasympathetic. So it's a nice figure, but don't miss that the spinal cord ends higher than you think. There are a lot of psychology textbook books that get this wrong. I bet some of your books in high school got this wrong too. They have the central nervous system ending down here, like you're sitting on your spinal cord. That is so not true. Um, Nucleus, collections of neural cell bodies. Um, of course, there's also a nucleus in each cell. And collections of neural cell bodies in the peripheral nervous system are called ganglia. I forget whether this book mentions that term. And there's the spinal cord and sensory and motor. And the spinal cord is really thin, thin as your little finger. There's not much to damage there that doesn't destroy everything. And there's the brain. And if you're my age, you know that movie. If you're your age, you probably don't. Young Frankenstein. What's really Frankenstein? And there's the outside of your brain, cerebrum. The outer part of the cerebrum is the cortex. And the brainstem. I'm a traditionalist. I like that as two words, brainstem. You can see the brainstem more clearly over here. This, what is he doing? I didn't look this up. He seems to say the brainstem stops at the midbrain, but he's colored the thalamus the same color. This is actually inconsistent even in good textbooks. That is, whether the thalamus is part of the brainstem. I kind of like to think that it is. By the way, this indicator is not pointing to that circle. That circle doesn't even exist in some people. It connects your left and right thalami. So don't get confused. The thalamus is quite big. The hypothalamus, like hypothermia, is below the thalamus. And cerebellum, notice, is purple because the cerebellum is not part of the brainstem. The spinal cord is not part of the brainstem either. But you'll often sometimes see the cerebellum mistakenly included in the brainstem. That ain't the case. Again, I prefer brainstem as two words. And there's thalamus, there's hypothalamus, there's the cerebellum, cerebral hemispheres, which we saw in chapter 1. And I said, don't worry about learning them over there. Cortex, outer covering or rind. Cortex is thin. It's in us. It's convoluted. I like to think of the cerebral cortex as, or the cerebrum as sort of being like a walnut, and it's connected. The left and right halves are connected by the corpus callosum, and we've got lobes. And this should look familiar from chapter 1. You should know these lobes and basically what we do with them. Temporal lobe is going to come up a lot because the amygdala is buried in there, as is the hippocampus. They're easy to see when they are color-coded and have lines around them. When you're looking at the real brain, you have to kind of know what you're doing. Prefrontal cortex is way important. It's a subdivision of the frontal cortex, of course. And particularly if you're a male, your prefrontal cortex hasn't fully developed yet. And that means your ability to engage in impulse control hasn't fully developed. It's not going to happen until you're about 25. So when you have impulse control problems, Uh, try to be chill. Broca's area, I'm not a fan of Broca. He was a racist, sexist, but we saw his area colored in chapter one, and it's important for speech. No doubt about it. Left uh, hemisphere in most people, even if you're left-handed. And then there's parietal lobes and somatosensory cortex and how we map them and homunculus. I always used to say in class that I wanted one of these, and a student thoughtfully got me one. It's a little statue in my office, and it looks distorted because it is, because the size of the cortex that's devoted to a part of the body is based on how complicated the control is of that part of the body. And you can see the hands are giant, and the tongue and lips are pretty enormous. The rest of it is largely along for the ride. Occipital lobes, visual cortex, temporal lobes, more on speech in there. By the way, that's not Wernicke's area, that's Wernicke's area. Again, German, from Germany. And you should know that the motor cortex is in the frontal lobe, and the somatosensory cortex is actually in the parietal lobe. Parietal lobe, right here, somatosensory cortex, motor cortex, somatosensory cortex. And there we have a lot of interesting stuff that's very relevant to clinical psychology and neuroscience. Basal ganglia, important for motor control, relevant to Parkinson's disease, and the limbic system, emotions, some forms of memory. And I mentioned that the amygdala and hippocampus are in the temporal lobes. And there's a summary, which is quite good. And, there's that the right side of the cerebral cortex deals with the left side of the body and vice versa. So if you have a stroke and you're paralyzed on the right side, and it involves the cerebral cortex, the stroke is on the left cerebral cortex. And peripheral nervous system. And we have nerves and the body senses, the autonomic nervous system, sympathetic and parasympathetic. It helps me to say the sympathetic isn't. It is involved for doing stuff and keeping you alive in fight or flight situations. It's good to have it, but it's not good if it runs all the time. It can be destructive if it runs at high levels all the time. And this is one of those figures that you shouldn't worry about, but there's your sympathetic and your parasympathetic. What it does to each side. The vagus is huge these days. They're stimulating it to treat depression, among other things. And there it is, slowing the heart rate, and you should know what neurotransmitter is being released there. Yes, it's acetylcholine. And there's the parasympathetic nervous system. And how do we look? I've done a lot of lesioning in my day, knife cuts and electrodes, lobotomies, knife cuts. I've always wanted to experience transcranial magnetic stimulation. They were selling these things at the neuroscience meeting I go to, and I said, do it, do it, do it, but their lawyers wouldn't let them. So I've never experienced it. It's quite safe. EEGs, I've had a lot of those done I help one of my friends do his research. You get this crusty white stuff in your hair all the time. And there we are watching things that to me really looks like single unit recordings, not EEGs. But mirror neurons have become, for a while, were an obsession in neuroscience. And they were proposed as being important for autism. I'm not sure. I'm not sure. I haven't invested heavily in mirror neurons yet. And there's different kinds of scans, CT scans, which usually don't look that sharp. Give me a second here. I think he's reversed these. Notice how detailed the one on the left looks. You can see white and gray, and I can see the thalamus, and certainly we can see that this person is in trouble here. Over here, things are murkier, and usually there's much better resolution with MRIs than there are with CAT scans. I think those two images are reversed. PET scans, I think you can get a PET scan in Scranton now, and they're used for research and some diagnostic purposes. fMRIs, functional magnetic resonance imaging. So they're looking historically often at blood circulation in different parts of the brain with the idea that if neurons are active, you need more blood, and it's true, and there's more oxygen in those areas, and they can light up. But it looks like it's a very detailed picture, very anatomically rich, but it's not. It's a functional MRI, and MRIs are detailed. So you get a free MRI when you do functional MRIs, and you can take your MRIs and make them into nice pictures. I have a collection of MRIs. I used to get them all the time, but haven't had one in a while. PET scans. I almost stayed at UCLA to do research in this, but we didn't think we would get the grant, but they did. But I was gone. And what we can learn from different kinds of scans. And here's how we do research with it. And computers are vital to this. There's a different image where you take a baseline without the stimulus you're interested in, and then you collect data when the stimulus is there, and the difference between those two indicates what's been activated, often indicates what's been activated by the stimulation. Neuroplasticity is important. And we're always plastic. If you can learn, you're plastic. But generally, as you get older, you become less plastic. And again, particularly males, you're still trying to get your prefrontal lobes and your impulse control under control. So, certainly our environment, our learning environment, our socioeconomic environment, all those things can affect the development of the brain and future behaviors. Whoa, connectomes. This is like magical. And those pathway maps are unbelievable, what they can do with these scans. These largely came into existence as I stopped doing research that involved anatomy. But wow, is that stunning. That's not, the picture on the bottom is what I'm wowing about. That's not, I mean, it's colorized, but the details are really there. And yes, diffusion tensor imaging. I've never actually held one of those in my hands or looked at one directly, but geez, I would pay good money. And we're out of here. Again, be able to use this Muse system on every chapter. And of course, the M in this is really, really relevant. It's all relevant. The unconscious, social affecting the brain, experience affecting the brain. I'm a big fan of neuroscience. And I don't know if there's anything here that we need to worry about. But look at the number of terms in this chapter. And if we weren't having an open book test, you'd have to know all those to be able to define them. And you still may have to define them, but it's an open book test. So thanks for stopping by. These are all the lectures you need for your week one, if I'm not mistaken. No, there's one more. But the quiz for the fourth lecture is next week. So one of these is for the first day of class, Tuesday, Wednesday, Thursday. This is the last for week one. So have a good weekend.